So I'm going to be talking about floral tube length divergence and how this can potentially result in reproductive isolation. Okay. And I'm going to be talking about this system over here, which is a plant called Laperosia anceps. It's a plant in the iris family, and it's pollinated by these long proboscis flies, which can vary in their tongue lengths across the landscape. I'm going to contextualize this work within the body of literature on ecological speciation, and I'm going to divide the talk into two broad parts. The first part is going to lean very heavily on one of my colleagues' work, Anton Poe, and it's going to show you how fly pollinators have driven the divergence of floral tube lengths. And the second part is going to detail the mechanics of how floral tube length divergence can result in reproductive isolation. Obviously, when you think of ecological speciation, you might think of examples like this, Patrick Nozzle's work on Tamima walking stick insects. But in fact, pollination biologists have been thinking along these lines for a long time, probably since the 60s or 70s. And I'd like to just put up this paper over here by Grant in 1994, which suggests that divergence in response to different pollinators can drive the formation of isolation barriers. And he's recognized two different ways that this could happen. The first he called ethological isolation, and here he suggested that, well, when flowers diverge phenotypically in response to these different pollinators, they end up attracting different pollinators, and so that would obviously be an isolation barrier. And there's quite a lot of evidence to suggest that that is the case in many different examples. The second one over here he called mechanical isolation, and here he suggested that adaptations to different pollinators can end up causing pollen to be placed and received on different parts of the pollinators. So even if those pollinators move between these divergent phenotypes, they don't move pollen very easily between those phenotypes. And this has been very poorly studied, and that's going to be the subject of my talk today. And I'm going to be talking about it in the context of this system. So here's this Laperosia anceps flower, and you can see over here a map showing the distribution of pollinators that pollinate this flower, and you can see three different fly species here. You can see their enormously long tongues, so the different color bars here are different color fly, different species of fly, and each bar represents a population, and the length of the bar represents the length of the fly's tongue, the average length in each of those populations. So you can see that there's variation between species, but over here you can even see variation within a species of fly. So if you look at tube length plotted onto this picture, so these gray bars now are floral tube length, and I've just plotted them for that one variable species of fly. You can see that the tongue lengths of the flies and the tube lengths are highly correlated. And if I plot in these other two species of or these other populations pollinated by the red and the blue fly, you can see them. Where have they gone? There you go. You can see them on the graph now. So this very tight association between tube length and tongue length perhaps suggests that floral tubes in each of these populations has adapted to fly tongue length in each of these populations. And to lend some more weight to that observation, Anton Poe in 2009 did these really neat studies where he showed very strong pollinator-driven selection on floral tube length, where floral tube length was selected to evolve similar to tube lengths to the pollinator's tongue lengths. So these data suggest that ecological divergence has occurred in this species, and that floral tubes have diverged in response to these fly tongue lengths. But what I want to talk to you today is really about reproductive isolation. So how does this tube length divergence result in isolation? And so back in 2003, Spencer was on sabbatical in South Africa, and the two of us were walking around, and we came across this marvelous population of plants, Laperousias, which was bimodal in its tube length distribution. And we looked at this population, and we've been studying now this population for the last 15 years. We've only ever found one species of fly in this population. So there it is, a picture. And its tongue length matches beautifully with this peak over here. So it's about 50 millimeters in length. And so it suggests to me that this population historically was a long tube population, and we think that some short-tubed individuals have moved in from somewhere else, colonized, and managed to persist in this bimodal state for the last 15 years. So how they managed to persist? Well, for it to have occurred over 15 years and not broken down yet, there's probably some kind of reproductive isolation happening. And we wanted to know in this particular study whether mechanical isolation was partly responsible for that. So as part of a study in 2016 with Spencer, we engaged in the long and forgotten art of allozyme electrophoresis. I wanted to call it a dark art. In this study over here, we collected seed progeny from short and long tube plants in the field, and we from this bimodal population, and we looked at allozymes, and we found complete allelic frequency differences for two loci over there between the short and the long tube forms, and for another two loci we found nearly complete differences. So it suggests that there is very strong gene flow barriers between these two forms. And we know that these forms are cross-compatible, so they do form some seeds. In fact, quite a lot of seeds when you cross them. So the question now is whether this mechanical isolation is partly to blame. And do we find plants receiving pollen on different parts of fly pollinators? So to answer this question, I collaborated with my two postdoc students, or I shouldn't call them students, postdocs, Cornel Minnar and Marina Stoyaga. And Cornel in particular was instrumental in helping to find a way to mark and follow pollen grains. So he did this by using quantum dots, which are these fluorescent nanocrystals the size of viruses, and they can fluoresce in different colors. You can apply them to the open anthers of flowers. They mark the pollen grains with different colors, and then you can pick up pollinators which have visited those flowers, and you can find those pollen grains on them. So there you can see those fluorescent grains. You can also follow these pollen grains to the stigmas of the flowers that the fly has visited. So then what we did is we asked, well, do short and long tube flowers place pollen on different parts of the fly? So we marked short and long tube flowers with different color quantum dots. We allowed these pollinators to visit these flowers. We then captured those pollinators and looked at them under the microscope to see where the pollen was placed. So you can see we don't have massive sample sizes because we didn't want to kill too many flies. But this is more or less what we found. What you can see here is that long-tubed flowers are placing pollen almost exclusively around the head and upper proboscis region, while short-tubed flowers, it's quite interesting, place quite a lot of pollen down here where you expect these flowers to pick it up. But they also place pollen sometimes over there. Short-tubed flowers were not very good at placing pollen, so we had lots and lots of zero placements from short-tubed plants. And you can see here that long-tube plants per visit placed about 3 times as much pollen as short-tube plants. Okay, so now what we wanted to do was figure out whether this weird placement of pollen results in a sort of pollen movement. In other words, is there more pollen movement within morphs than between? So you might think that, well, if morph ratios are equal, if pollinators forage completely randomly, if pollen is placed in the same part of the pollinator, you'd get long tube plants receiving half of their pollen from other long tube plants and half of their pollen from short tube plants. And similarly, you'd expect the same for the short tube plants. But in reality, we know that these flies are carrying pollen in a weird way, so how do we figure out what's going on there? So what we did is we designed a completely new experiment. We stained short and long tube flowers with quantum dots of different colors. We allowed these flowers to be visited by these pollinators. We then allowed those pollinators to take that pollen and transport it to a receptive flower, and that flower would either be a long or short, it didn't really matter. But once we had collected those data, we then harvested the stigmas of those flowers, looked at the pollen grains and had a look to see how many grains had been moved. So using those data, we were able to model pollen movement under conditions of completely random foraging and under equal morph ratios. And these are the results we got. So the black lines are what you'd expected, what would you'd expect given a 50-50 sort of pollen movement. The colored lines are what we actually got. So what you can see here is long tube plants. So the thickness of the arrows is relative to the amount of pollen being moved. So you can see that most of the pollen is being moved from long flowers to long flowers. Long flowers are receiving 80% of their pollen from other long flowers, only 20% from short. Short tube plants, you can see by the narrowness of those arrows, are moving very little pollen around. And even though they're moving very little pollen around, 96% of the pollen found on short tube stigmas was from other short tube flowers. So to sum this up, if you just look at tube length as a reproductive isolation barrier, long tubes are about 80% isolated, short tubes about 94% isolated. If I add in other reproductive barriers that we found in this study with Spencer Barrett, the reproductive isolation becomes much more intense. Long tube flowers are about 98% isolated. Short tube flowers are about 100% isolated. So that probably explains why this bimodality has persisted for at least 15 years. We don't know what's going to happen in future, but it's persisted for this long. So just to end, I think we've managed to show that tube length divergence can result in reproductive isolation, and that's because these different placement and receipt areas result in pollen being received differently by short and long tube plants, and that results in isolation.
So style length and pollen size often co-vary. Do you see any of that?
Sorry, does style co-varies with
Pollen size?
pollen size. There are very, very slight differences, and we wanted to use pollen size to distinguish pollen grains, but it's not that good that you can do it.
So I'm
There's
just
quite a lot of
wondering if
overlap.
maybe the pollen from the short style morph, even though it gets often to the long style morph, actually doesn't sire seeds.
So we actually did that in the 2016 study, and the interesting thing is that it was the other way around. So the short styled pollen was better at siring seeds on the longs than the longs were at siring seeds on the short. So it was not what we thought.